Greetings, I'm Yvonne Stapp for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Today, we're getting an update on the technology of brain signal recordings, electroencephalographs, or better known as EEGs, and the medical devices they can be used for. Our guest is Dr. Shawn K, Senior Systems Scientist for Engineering Research Accelerator and also Courtesy Professor for Electrical and Computer Engineering at Carnegie Mellon University. Dr. Kelly has developed a number of medical devices, including an implantable retinal prosthesis, an artificial eye for the blind, which he described some years ago on this program. And I've asked him just talk a little bit about that again very quickly. His current project of interest is a technology to improve EEG recordings and what they're used for. We're about to learn first what makes EEG recording complicated, and second, we'll learn about some innovative medical devices based on EEGs. Dr. Kelly, it is a pleasure to welcome you again. Thank you very much for joining us today.
Thank you so much, Yvonne. It's great to be here.
And to start with, could you tell us just very quickly a little bit about the work that we knew you for, about the implant, the retinal implant, just like a quick description, and where is it?
Sure. I spent almost a quarter of a century trying to develop a retinal prosthesis to restore sight to the blind. This is an electronic device that would attach to the outside of the eye and have electrodes in the retina in the back of the eye, primarily for people who have lost their vision because their rods and cones have died off through disease. And of course, the rods and the cones translate optical signals, light signals, to nerve signals. When they're gone, you have no vision. So we developed an electronic system to replace that function. A camera, maybe worn on a pair of glasses, would collect the image information. It would electrically stimulate through this implanted device the nerves at the back of the retina, which were still healthy and are still around. And that would send signals back to the brain to give kind of a rough pixelated version of vision. It wouldn't be quite natural. We were developing a 256 channel version of that. It's pretty high density. And we worked on that for a number of years. The funding just started to dry up. We were having trouble with one or two engineering pieces, and we've tabled it for now, but it is still holds a dear place in my heart, and I hope that I can get back to it at some point if we can raise funding again.
I just wanted to thank you for telling us a little bit about that, and we'll point to your interviews and stuff for this program in the past on that, but it got a lot of attention at the time. And since you mentioned the funding, we're asking scientists to talk a little bit about this issue, which is a real issue anymore. People really get stuck. So we'll take that up again a little bit at the end if we've got time, and if you would be so kind. Because
of course,
we in the public really need to know about this, and it's not out there in the media, the issue of funding and how people just get, sidetracked with their projects. In any case, that now we'll go to the EEGs. And so in this kind of thing that you're working on, how do these EEG recording devices function, actually? I think people are familiar with them in a vague way.
Right. So traditionally, EEG puts a number of electrodes, typically between 19 and 22, depending on which extra electrodes you add, across the scalp. and they are applied as individual electrodes that are very near the skin and with a little bit of maybe conductive adhesive paste that sort of holds them down to the skin all throughout the scalp. There's a whole range of them. They've got wires that then connect over to an amplifier system and digitizer system. And the data goes into a computer, and you can look at all these different channels and look at brain waves, broadly speaking. Now, this is on the scalp. So you have to go from the brain through the skull to the scalp, and there's some amount of distance between the activity in the brain and the electrode on the scalp, but you can still get a lot of bulk brain activity information from this non-invasive, completely non-invasive measurement. Just briefly, I was talking with some colleagues in neuroscience and some colleagues who do some signal processing work, and the colleagues in neuroscience, including Professor Marlene Behrman and Professor Michael Tarr at Carnegie Mellon, they thought, it would be nice to be able to get more information, to try to get higher densities of EEG systems. And my colleague Pulkit Grover at Carnegie Mellon said, no, I think there's probably more information there than we're pulling out in 20 electrodes, which goes counter to arguments that have been made in the past that the skull acts as kind of a filter, kind of low pass filter spatially, and it squashes a lot of information from getting out from the brain to the scalp. And so 20 some electrodes is all you can do. And we've tried to push that envelope and see how much more information we could get.
I see. And so it picks up the brain waves. This question of how much information it can get. So that's one of the concerns. And then also, could you tell us what some of the uses or devices the EEGs serve? We think of it as diagnostic, I think, in general. That's the thing that we're most accustomed to. But can you tell us anything about that and others?
Sure. It is generally a diagnostic tool. The biggest use is in epilepsy studies, finding seizures. A seizure is a giant storm of electrical activity in part of the brain. And so if you're measuring bulk electrical activity from the scalp, you're not measuring individual neuron spikes or anything. But if there's a giant electrical storm, you can easily pick that up. So that's what it's been used for, is finding seizures, trying to localize seizures, which is difficult to do, trying to diagnose epilepsy from patterns of seizures, things like that. It's also used in sleep studies. You can tell when the brain is active, when the brain is quiet and going into its rhythms of sleep. It can be used in some other things as well. It can be used in sort of studying traumatic brain injury. It can also be used in an emergency department when you have someone in a severe sort of brain injury situation, or even in other kind of emergency department situations, that you can have seizures that just sort of come out of nowhere and happen. So often it is used for long-term monitoring in an emergency department, just to pick up these seizures that are pretty small and would otherwise be missed. So there are quite a large number of applications and indications for EEG.
Right. And just very quickly, I would imagine, because is this used for stroke diagnosis also by any chance? People seem to have those symptoms that they can track that.
That's a good question. It can be. It's not widely used for stroke, but it is. I think it's gaining acceptance, and there are some algorithms that are being developed for stroke.
I see. That's one of the things I imagine you're working on, is the algorithm. So that's very useful. I don't know. Also, is this involved in like brain-computer interface devices or mechanisms now, where you're giving an opportunity for, say, what we call locked-in people, I guess, with really paralyzed and can't move anything, but their brain waves can direct mechanisms, computer mechanisms. Is that part of this too?
Yes, that is the hope. It's difficult, and we are not there yet.
Yeah,
Currently, these brain-computer interfaces for these sorts of people involve very invasive, penetrating electrodes that go into the brain itself,
yeah,
um, and then you can individually record neurons that are spiking, and you can interpret that information to
right.
try to control things. It's very invasive.
Yeah.
It turns out not to be a very stable system. You put electrodes in there and your body doesn't like them, and so your
Ah,
body attacks them. They only work for weeks or months or maybe a year, and they start to fail. Um,
ah,
and it's also just invasive. So we're hoping that there's a way to do that, not by recording individual nerves, but by recording broader
yeah.
patterns of activity from the scalp with EEG. Again, we're not there yet, but it is something that we are working toward.
Right. Is this part of your current work, your current project, this getting at the algorithms or making these things have greater capacity?
Greater capacity, yes. Brain-computer interface is something we're thinking about.
Yeah, yeah, no, I mean,
Yeah, so the current work on algorithms is more on the diagnostic side still,
yeah, okay.
trying to expand EEG into other diagnostic areas, uh, just with much better algorithms, much better diagnostic tools.
Right, I see. So here is the thing. I think I didn't realize that it's limited. You think that you put that cap and all that goo on the scalp, it ought to tell you a whole lot. But apparently, there's a real need to improve that technology because it could help out so much. I guess with diagnostics in particular. Um, what are you doing in that regard? How do you go about that? Here's the problem. It's not
Mhm.
completely efficient and you're trying to improve it. What's your project there?
So let me give one particular example. If someone has a severe traumatic brain injury, they're in a car accident, there's military, and there's an explosion nearby,
Yeah.
whatever, or sports injury. Um, these patients come into the hospital and they're stabilized, and then there's a bifurcation, there's a fork in the road. Either they're going to get better on their own, or they're going to get worse.
Mm,
And healthcare finances being what they are, you need to make the right decision. You would love to just keep everybody in the hospital for weeks on end for observation, but those beds are needed by other patients. A physician needs to decide, is this patient going to get better? Let's do that at home and open up patient resources and beds for other patients. Or does this patient need a lot of care? In which case they need to be held longer. There are drugs that they can be given to kind of quiet things down and prevent ongoing sort of secondary brain injury. And there's no good way of knowing. Um, One way that you can find out is there's a biomarker called cortical spreading depression, sometimes cortical spreading depolarization, CSD. And the CSD is a wave of sort of depressed activity that moves very, very slowly around the brain. Um, and you can measure that with electrodes on the brain surface, on the cortex. So you take off a piece of skull, put electrodes on the brain, and a clinician then looks at all the EEG recordings and time sequence, all the recordings in a row, and through a very manually labor intensive process,
mm,
can determine that this patient has these cortical spreading depressions. Um, it's very invasive, it's very time intensive. What we're working on right now is finding a way to do that from the scalp, not from the cortex surface. So
mhm.
way up, non-invasive. And secondarily, to do that as an automated process. And we've done that. We've determined that we've found these waves of depressed activity moving around automatically from scalp electrodes, which is a big improvement. This gives the doctors now information that says, this patient is going to get better, let's send them home. This patient still has this propagating wave. Let's keep them in for observation. Let's try some drugs that we wouldn't normally try to prevent that secondary brain injury.
Ah, I'm beginning to see it now. Now, here's the thing that you say that you're doing this on the scalp. I'm thinking the EEG, that cap that they put on and you put this glue and so on. A couple of questions there is what you are doing different when you say it seems like whatever you're doing is much more efficient, but I don't know if it's the same procedure where you put that calp on and you put all that glue in there and stuff.
Well, we've got a sort of two different parts of our system. One is the algorithms, and so far we've tested those with exactly those electrodes you're talking about. Somebody applied the electrodes with the glue and the smelly, sticky stuff. And there were 21 electrodes.
Yes.
And we took that limited set of data and we used those data to test our algorithm, and it worked very well. What we would love to do is take our algorithm and apply it to a much higher density set of data, right? With maybe hundreds of electrodes on the scalp. Here's the problem. Placing those electrodes on the scalp takes 25 to 30 minutes for 21 electrodes. How long is it going to take to place 250 electrodes on the scalp, right? That's going to be a while. So we're developing a cap that has clusters of electrodes, and this is more of a quick apply cap. We're developing new types of electrode materials, new types of electronics, all of it built into a mechanical cap that can drop into place. And that allows you to scale up the number of electrodes.
Right.
And that allows you to give the algorithm those data, those very high density data that allows the algorithm to work even better. So we've got sort of a hardware and a software component to what we're developing.
Yeah, I see. I have one more thing here. I understand that apparently, what shall I say, some scalps, more particularly some hair, is not as receptive, that it makes it extremely complicated to actually get the electrode onto the scalp. And that so hair of different types is uncooperative, or is that an actual problem? And the other is that, as you said, it takes all day to get the thing attached.
Mm-hmm.
And then it's a mess to get that stuff out on top of it, the gluey, whatever it is, adhesive. Could you tell us a little bit about that? Because that was new for me the first time that I saw that actually some hair is not cooperative. It gets in the way.
That's exactly right. And it's a problem that's kind of partially recognized in the field. I think there are plenty of clinicians out there that don't quite see it as a problem, and others do. So I think we're working on trying to build awareness there. We actually had a student named Arnell Etienne, who's now working for us. And she found out about this problem, learned of this problem. She herself has a lot of coarse curly hair. This is, sort of a common thing. African Americans,
Italians,
sometimes, yeah. With a lot of hair,
thick curly hair.
Yeah, so there's thick hair, and there's also the special springiness, and there are sort of gradations of this, and particularly African hair though does have the extra kind of springiness that pushes back. And when you put these electrodes in place, you sort of part the hair, try and put these things down, and the hair just really pushes back hard. And so in both clinical EEG and also in research, EEG is widely used in research to study a number of different things. In clinical EEG, you've got problems where either the EEG doesn't work or the technician comes back over and over to replace an electrode here and there. In research EEGs, sometimes researchers have to say, I'm sorry, this subject just doesn't work for our study because the hair is going to be too difficult. And now you have a study that's not very representative of the population.
And people don't want their scalps shaved. Is that the idea?
That's the thing too. You bring in your child with this big, beautiful head of hair for an EEG, and the clinician and the technician look at that and they're trying and they say, we might need to shave your kid's hair, and nobody wants that.
Nobody wants it.
So what Arnell worked on was trying to find ways of leveraging the hair, and of course there's sort of a cultural tradition of braiding, all sorts of different styles of braiding.
Right.
And so she looked into, can you braid to open up the areas where we need electrodes for this sort of so-called 10-20 electrode system? And she worked on that, and that helped a lot. And then she invented a type of clip that kind of slides under the hair,
Mhm.
and this is a patent pending technology. It uses the hair's strength to hold down these electrodes.
Oh,
And we're actually starting some clinical studies at the University of Pittsburgh within the next few weeks.
yeah.
It's trying those out. So it's a really
So
great tool that helps make EEG more accessible.
yes, but because I guess that would really mess things up if the hair keeps getting in the way, and we never think about that.
Mhm.
But if you were going into the hospital and had to have an EEG, and they said, "Could we please remove your hair?" It would just complicate things. Now, in the other thing is, it sounds like you're making, is it possible, a more complex array of these electrodes? Is that so that you're getting more? Is this, uh, can you tell us something about that when you talk about it's getting
Sure.
more?
Yeah, so we're starting by developing a cap that has the standard number of electrodes, the
Yeah.
21 electrodes. Um, and that will just serve to simply speed up the process, which is important, right? It takes
Yes.
25 to 30 minutes. You just drop this cap on, adjust it, 5 minutes later, you're ready to go. Um, for the healthcare system, that's a bit of a cost savings. For patients, it's a bit of convenience. For some patients, it's a big deal. I work with pediatricians, pediatric neurologists, and the kids who are coming in for EEG often have other medical complications.
Ah,
And these are kids that have other kinds of things going on. They don't want to be touched. They don't want to have this person putting these
yeah,
things on
yeah,
their heads.
yeah.
The smells, the sounds are very triggering. Um,
Oh,
and they can lash out. You can actually have staph injuries
yeah.
from trying to put an EEG system onto an uncooperative kid.
Yes.
And so we think that that's a case where this quick apply EEG cap will really, really make a big improvement. It'll make things much easier. 5 minutes of adjusting a little hat, and they're done. But as I said, that's also a requirement if you want to go to very high densities. You can't place individual electrodes. We're then going to modify this cap to have clusters of electrodes, clusters of amplifiers, and be able to use the same basic hardware to scale up to 64, 128, maybe hundreds of electrodes. And this then allows us to get much higher density for that algorithm I mentioned for traumatic brain injury, but also for research. If
Right.
it's true that
Right.
there's more information in EEG than we're actually pulling out with 21 electrodes, then if we have a cap with large numbers of electrodes pull out a lot of data, then that opens up the world of clinical research. Let's see what else is in
Yes.
there. Let's dig
Right.
through these data and see what else we can determine.
Right. Now, I was just thinking of like like functional MRI, you just slip this person in there, but is that not as good as EEG because it does seem like EEG gets a lot of use for damage
Mhm.
like this?
There are two major differences between EEG and MRI in general, fMRI in particular. One is cost. MRI is an expensive tool. EEG is a
Yeah.
much, much less expensive tool. Um, the other thing, though, is temporal resolution. MRI gives you amazing spatial resolution deep into the brain.
Hmm.
You can see incredibly
Hmm.
small areas, and it's great. But if you want to do kind of an fMRI and study some stimulus and study the brain's response, you don't have as quick a time response if you're studying it with fMRI. With
Oh,
EEG, you can get almost instantaneous temporal resolution. As fast as the brain can respond to what it's
mhm.
seeing or doing, the EEG can record it. Electronics are much, much, much faster than your brain. The fMRI is just a slower machine in terms of trying to see quick responses of brain function.
Yes, I just incidentally, I would imagine that if you had a brain injured person or child in particular who had other difficulties, that confinement and being slipped into this coffin like device, a lot of us wouldn't be able to handle that very well. But
Yeah.
a youngster might have real problems. So that would be another thing. I didn't realize that the EEGs, though, are much more really supply much more instant information for you. And I can immediately see the research possibilities too. But to clarify, that you still need to put the glue on that scalp. Is that it? And put those, like increase the number of electrodes.
So for the quick apply cap that we're developing, there wouldn't be a glue. There would be a
Oh,
cap that would be doing the kind of pressing that would take the place of adhesives. It would be a little wet, we think, is the plan. People have tried dry electrodes, just metal electrodes touching the
yeah,
scalp, and the quality just isn't quite there.
I
So we're
see.
looking at kind of a sponge type electrode that holds its moisture for a very long time, that does work dry, we think, with conductive electrode materials. But the moisture that would be on the scalp would be water or saline. There wouldn't be
Oh,
kind of
you
the
don't have that
goopy,
awful,
gluey, no.
yeah. Yeah, that's very interesting. So you get more with
Mhm.
simpler mechanism in a way there. So that's one thing. Could you tell us a little bit about the process of developing these kinds of things? You've developed a number of things, and that it always with these wonderful innovations. They take a long time, and they take a long time apparently to go from the creation to patent to whatever, but you give us an idea of what the time is in terms of developing something so important as this.
Sure. So in the engineering field, you come up with an idea. Maybe you're working with some scientists. You get some inspiration, and then you just have to kind of prove out the concept first. Very early on, you're kind of, I would say, de-risking, right?
Mhm.
You're making sure that something functional is going to happen, and is it worth pursuing all this extra work. And so you put together something very simple in the lab, try and make it work, and then you iterate and you build. And this is a case where some of the modern tools like 3D printers are very nice.
Mhm,
Now
mhm.
you want to have something made, you design it on some software and you send it
Yeah.
to the printer, and you can have something in a few hours. You can
Wow.
have a lot of pieces in a few days. Also, the electronics industry has made making little printed circuit boards, custom designed circuit boards with components on them. That's become faster, easier, cheaper. And so, you can make a custom circuit board every time. When I was starting out, if we wanted to try a new circuit boards were expensive, so we would wire things up and run wires everywhere and solder everything, and the process would just take longer. Um,
Mhm.
Now you can get a circuit board back in a week, shipped to you
Oh
from
my
somewhere, and
goodness.
it's totally put together and it's totally custom built and it's flawless. So that helps too. So there's definitely some technological advances that help this process, but it's still make a simple thing and then test it. And the testing phase takes some time. You have to
Yes.
collect the data, analyze the data, make sure that your electrodes have good resistance connection to the scalp. Um, Things like that. Make sure that the EEG waves make sense to people who look at EEG all the time.
Yeah.
Um, and then of course there's always the funding issue. You do a little bit of work and then you use that work to apply for some maybe small grant. You get the grant, use that money to do work, make sure you do enough more work that you can have preliminary data to apply for a bigger grant, and then trying to leverage that up to where you can have enough money to really run the operation, um, and get things done in a timely manner. The patenting process at a university, it's kind of nice because you come up with something, you just report it to the university, they talk to you a little bit, they file the patents for you.
Ah,
That process is easy. Of course, then if you think that you want to commercialize that, you need to go back to the university because they then own those patents, and you need to license them back if you wanted to form a company and actually make these things and make products. But
Yeah.
still, that's a nice system to have as well.
A couple of things. First of all, on that funding, thank you for that, because it's important that the public understand that scientists are running around all the time looking for money, and that they can really get stuck, because you're in the middle of something important, but you get, if you're lucky, a 4-year grant or something like that, and then you're stuck. And we've had recent years cutbacks in important areas. And so it's important that the public know that we would like our tax dollars to be sort of used for research and in an effective way. The other thing you brought up was the patent thing. So now we understand that the universities take care of that little item a little bit for you. And then another thing is that people, innovators like you, very often go into startups. So here in the Cambridge area, the place is crawling with startups, sort of, especially biomedical, the field. Well, are you going to consider a startup for your work also? And if so, would you tell us about it?
Absolutely. So going back to the earlier discussion of the retinal prosthesis, we did begin a startup company for that. It
Ah,
turns out that it's very difficult to raise money for that.
yeah,
It's an expensive project,
yeah.
um, and the market is maybe not big enough, and there's enough risk that people were a little wary there. We're still working on that. We're still pushing. We think there's some real promise there. But in the
But there's a
shorter
real
term,
need for it.
I
Yeah.
think so. Yeah, absolutely. In the shorter term, this EEG system, we are starting a company. Um, it is going to develop both the short-term quick apply cap as an early product, but then also this cortical spreading depolarization brain injury
Mhm.
studying system. And so we're currently trying to raise investment capital for that. We're applying for, again, grants. There
Mhm.
are small business grants that can help get a business off the ground. So we're in the process of both raising grant funding and raising investment capital for that. And the thought is that we can sell some of these EEG systems, including, by the way, that first clip for coarse curly hair for EEGs. That will be part of the product line as well.
That is terrific, because I can see that there's a great need. I just am curious if it's just more efficient for innovators to just do a startup so they can hurry up. And if they can get the funding, they can go ahead. But if you're running around on the other hand, and you're doing your research and you're going grant to grant to grant, and everything is uncertain, maybe you just get really frustrated after a time. I have no idea. But is that part of the attraction of doing startups? Because that's like another profession practically.
It is. It really, truly is. And I think it depends on what you're working on,
Uh-huh.
and then your particular inclination. There are plenty of professors working on very commercializable
Yeah.
uh, technologies that really enjoy working in the lab and doing research and teaching. And there are professors who are working on really important research that's either not ready or just doesn't translate to commercialization in the same way. I think we've gotten lucky here that we're working on a project that does have some commercial
Yeah.
value. I, in particular, am really excited about the idea of moving into a startup and working for a while, trying to build something, trying
Yeah.
to put together a company, put together a series of products, lead a team,
Yeah.
and get it out there. Um, I think any researcher wants their work to be, let's say, just out there, right?
Yeah. Sure.
Recognized, either publish things so that other
Right.
people have that information and can build on it, or really put a product out there in the world so that people can use the results of the research. And startup commercialization is just one way to really put your research out there in the world and have people use it.
Right. Well, it seems like in a case like this, with something that is so important, there would be a lot. It would be it was a lot of reception for this, a lot of eager reception because it's much needed. I'm thinking of all the brain injury kind of things, something that would give you the better information faster. That would be a real plus, and there'd be every reason to, I guess, what you say, commercialize it, but you have to be the right type of person to do it. So this is, I'm sure, an exciting thing for you. I'm glad you're an entrepreneur in addition to being an innovator. I don't know how many of those there are, but it's probably very exciting. I know you will be successful with this. I look forward to hearing more about it as it comes out to market. Um, Dr. Kelly, thank you so much for telling us about this very interesting development and the need for it. And all the best luck. And please come back and tell us more in the future.
Thank you so much, Yvonne. This has been a real pleasure.
It's been delightful to see you again. Thank you. Bye bye.
Thank you.